We're building technology to read the activity of all the neurons in these brains and to write activity to control and manipulate the activity of these neurons in the brain. And these technologies are necessary to be able to cure human patients. But the same technology is neutral. It could be used for good or for bad. The same technologies to read and write activity into brains can be used to decipher what people are thinking or to influence the activity of the minds of people. So you say, well, this is science fiction. Well, not really.
In the middle of the 90s, the last decade of the 20th century, and the second millennium, a whole revolution in the world of neuroscience occurred. The neuroimaging techniques appeared.
So neuroimaging techniques are important for this revolution in how to learn and explore the brain, because it's a nice, non-invasive way to get inside the brain.
One of the imaging techniques that had the greatest impact was magnetic resonance imaging. For the first time ever, we could see the inner functioning of the brain while it experiences sensations and emotions. In fact, functional magnetic resonance imaging, able to detect the presence of oxygen in the blood in the brain, has allowed us to discover something unimaginable years ago: how different areas in the brain interact while the patient talks, sees an image, listens to a sound, or simply thinks.
But if you ask him to focus on one task, like listening to a sound, only the region from this specific task, the auditory cortex, for example, will have more consumption of oxygen, and this region will pop out in the contrast if you analyze the data, listening to sound against not listening to sound.
The reason why we don't understand how the brain works is not because of its complexity, but because we've been lacking methods to study it at the right level. Now, if you think about it, trying to understand a system as complex as the brain that has 100 billion neurons by recording from one neuron is a little bit like trying to watch a movie in a TV screen that has 1 million pixels by looking at a single pixel. So no matter for how long you study a single pixel or you study a single neuron, you will be missing the figures in the screen and the shapes of the images because they are an emergent property of the correlations in space and time between the individual pixels. We think that neuroscience should embark on this period of new technology to develop the method so that neuroscientists and clinicians will be able to see for the first time in the future the activity of complete neural circuits and be able to decipher the functional meaning of this pattern. How can one measure the activity of each and every one of the neurons in a brain at the same time? The methods that we and many other people are using are optical methods. We use light because light can penetrate the tissue in a non-invasive fashion. In particular, the method that I pioneered is called calcium imaging. We used it for the first time to measure the activity of neural circuits. Every time a neuron fires, every time it's activated, calcium comes into the neuron. So that means if you have these little calcium indicators, these fluorescent molecules in all the neurons, you can directly see who is firing, who is being activated in the entire group at once, because you see these as changes in fluorescence that are happening in different neurons at different times.